Welcome everyone, and I hope over the next 20 to 30 minutes that I can provide you with an overview of Horizon's gene editing technology platform. I'd guess that many of you are involved in some way with the schematic that you see on this slide that depicts how genome sequencing data is being translated into better therapeutics and diagnostics. Genome editing is really just one of the emerging tools that's involved in that whole process. Horizon was started almost 7 years ago now to help take genomic information and convert that into useful information that speeds up the discovery of better drugs. Having the sequence information is great, but unless you have ways to interpret it, it really only has limited value. We use genome engineering to create cellular models of disease that help tease apart some of the subtle genetic differences that exist. We call our platform for gene engineering Genesis, and I'll be going into more detail about how we undertake genome engineering projects through application of a variety of technologies. So there are a number of ways that you can manipulate the genome, and these can be carried out in a variety of ways. The key question you should always be asking yourself really is, what's your goal? What's the question you're trying to answer? And is genome engineering going to get you all that you need? Although genome editing is really getting easier and easier, it still requires significant investment in time and expertise. So one thing we've learned is to truly consult our customers on just what they're hoping to achieve and make sure that genome engineering can deliver that for them. There are some very important considerations to keep in mind when you're planning a gene editing experiment. Among the important considerations is something that gets overlooked a lot, and that's really the cell line characteristics. The cell line that you plan to use for this needs to have certain characteristics in order for success to be achieved. One of the important characteristics, is the ability to propagate this cell line over time. So primary cells, at least currently with those methods that are available, are not amenable to this type of genome editing. The reason for that is you have to be able to propagate these over months. And the second point here, the ability to single cell dilute, is a key issue. None of the current gene editing techniques is really efficient enough that you can treat a population of cells and have enough of those to really give you a response that is indicative of the same sort of thing you would get from a clonal population. You also need to know if your cells will transfect or electroporate, particularly if you're planning to use nucleases. If you're going to use AAV, you need to know if they transduced with the virus. The copy number at the target locus is another important issue. And then the presence of allelic SNPs. If there are differences between the published genome sequence, for example, or even among the alleles present in your particular gene target, those are important factors that are going to play into the effectiveness of any gene editing approach that you take. And of course there's biology, and I think most of us take these into account when we start, but we don't always know the answers to these things. Will the change that we're introducing be tolerated? Is it survivable by the cell? Will the cells grow differently? Once you've introduced a genetic mutation, would you expect that population then to outgrow the cells that are still wild type, or might they be deficient in their growth? And can you compensate for that in your selection procedures? And then something that I think a lot of people overlook is, will the modified allele be expressed? You may be knocking in a point mutation, for example, and that's great if all you want to do is characterize the DNA itself. But if you need that protein to be expressed, that's an important thing. Sometimes modified alleles get shut down. So these are all important considerations when you're doing a gene editing project. Our platform, Genesis, is really devoted to trying to apply the right technology for the right situation. So what is it that you want to achieve? And what we like to think, at least here at Horizon, is that we're technology agnostic now. We aren't committed to using one particular technology for genome editing, but we will use the one or the combinations of those technologies which will give us the most efficient way of deriving at the right answer. What we currently have is we combine 3 different technology platforms: recombinant AAV, or adeno-associated virus. It's a high precision method, It can be used at any locus, and AAV itself, the virus, has very wide tropism. Greater than 90% of all mammalian cells can be efficiently targeted using AAV. It's well validated. It's been around for quite a while. We use it a lot for knock-ins, and I'll explain some of the differences, the advantages and disadvantages of these approaches. A second platform we use is zinc finger technology, or ZFNs. These are considered more medium precision, at least compared to AAV, because there is at least the potential for some off-target cutting to go on. Zinc fingers, however, have very good genome coverage. They are well validated, and they are a primary tool for doing knockouts. A third technology that we've started to employ recently is something called CRISPR. Many of you have probably heard of this now. It's more of a low to medium precision, at least that seems to be the case for now, although there are modifications to the CRISPR technology that seem to be improving it. One of the advantages of CRISPR is its ability to affect multiple genes at the same time. And it's very simple in its design. It's an RNA-directed system. So I said I would talk a little bit about some of the advantages, and I hit on some of these before, but for recombinant AAV, it's particularly useful for complex edits. So if you have widely spaced edits, things that are 20 to 100 to even a kb apart, then you want something like AAV that has about a 5 kb payload to be able to deliver that type of change. It's very high in its precision because it uses strictly homologous recombination, and there are no DNA cuts involved. Zinc fingers, as I mentioned, are particularly good for knockouts, disrupting a particular coding sequence, for example. They're also good at multi-allele targeting, and so if you have 3, 4, 5 copies even of a gene in your cell line, zinc fingers are one of the most efficient ways of going after them. Now, CRISPR, as I say, is a little bit new, but has most of the same advantages that you find with zinc fingers. It's very good at knockouts. It has also been shown to be very good at doing multiple genes at the same time. And I think one of the really key advantages of CRISPR, aside from just the simplicity, is the speed with which you can do these things, and that means fairly low cost as well. There is one technology maybe you might think missing from this. That's the TALEN technology. And really, with zinc fingers and CRISPR available to us, we didn't feel the need to bring in TALENs, but I would put them right up there and make them fairly equivalent with zinc fingers and CRISPR in terms of having a nuclease technology to undertake these types of things. So now I'd like to go in just a little bit in depth to each of the technologies, not too deep, but just to give you a little bit of an overview of each of them and how we're applying them and some of the improvements that have been occurring with each of them. So recombinant AAV is the technology that has really been at the heart of Horizon for about 5 or 6 years now. As I mentioned, it does not involve the introduction of any DNA damage. It is strictly using homologous recombination. AAV is a single-stranded virus, so what you get is a very efficient delivery of a single-stranded piece of DNA into the nucleus. And what is interesting about AAV is it seems to stimulate homologous recombination directly. So we will get about 1,000-fold higher efficiency in gene targeting than you would if you were to introduce the same piece of plasmid DNA. Excuse me. All of the alterations that are introduced using AAV are user-defined, right down to the single nucleotide. So you put into the vector itself exactly what you want copied into the genome. The wide tropism I mentioned before, about 90% of mammalian cell types are easy to transfect with this virus. But one of the drawbacks to AAV is it has a slightly lower efficiency compared to some of the nuclease technologies. It is particularly good at going after single alleles, but if you're looking at modifying multiple alleles at the same time, AAV requires that you do that sequentially. So its application for knock-ins and complex edits is very, very good, but it's something that we might not turn to as a first thought for a knockout. We have been working here at Horizon on improvements to the AAV technology. So one of the things that happens basically anytime you put a piece of DNA in, whether it's AAV or it's a plasmid donor or even an oligo, is that piece of DNA can get integrated into the genome either randomly or often goes in at the sites of cuts that are occurring within the genome. When that happens with AAV, one of the things that we've started to use now is a system that selects against that type of incorporation. So what we've done is to use shRNAs targeting endogenous genes, or in fact the marker gene that's introduced via the virus itself, way out on the ends near the ITRs. So when the whole vector itself incorporates into the genome somewhere, these shRNAs get expressed. And in the example you see here, these shRNAs would target that marker gene, which could be something like neomycin or hygromycin. And when you do a selection then, because these shRNAs have shut down that marker, these randomly integrated copies of the vector will not protect it. And so you can select against those. And we've got some results here down below showing how with the incorporation of a single copy of an shRNA on one side, we got about a 4-fold increase in targeting. And when we use dual shRNAs, one on each end of the vector, that we could raise that up to about a 10-fold increase. So this is one of the ways that we're improving our AAV targeting. Another thing we're doing is we're playing around with the mechanism inside the cell that controls homologous recombination. So there's an active DNA mismatch repair pathway that reduces the efficiency, essentially, of all targeted homologous recombination. And by suppressing a component of the mismatch repair pathway, we can improve the targeting efficiency. What we've done here is we've actually used an siRNA targeting a pathway, a gene in the MMR pathway, MSH2. And what we've seen when we've combined this, in fact, with the negative selection itself, is that we get an increase in the targeting efficiency up to 40-fold, as you can see down below, where we've got a single shRNA in the negative selection scheme plus the MSH2 siRNA. So we can realize pretty dramatic increases in targeting using AAV by combining these two new methods, the negative selection method and the mismatch repair suppression. Now in this nuclease pathways, zinc finger nuclease, as I mentioned, is part of our service offering. We've licensed this in from Sigma now, so we are actually using zinc fingers that are supplied by Sigma, the highest quality in the world. They are well validated for doing fast and efficient knockouts. So the one difference between using AAV, for example, in a knockout experiment and using a nuclease is the nuclease, the approach is to put the nuclease in and have it cleave, cause a double-stranded break in the DNA, which gets repaired through a pathway which is prone to errors. And you will often get small deletions or insertions of DNA at the site of the break. And what this leads to is a disruption of the coding sequence itself. So there's slightly lower precision when you're using this, but it is ideal for knockouts because of the efficiency increase that you get through this targeted cutting. It's easy to target 2 or even more alleles at the same time using a single reagent. And so it's not unusual that you would derive homozygous, for example, knockouts of a gene by using a ZFN approach. CRISPR works very similarly to how zinc fingers work. It's actually simpler to design. There's no protein engineering that's required. What is required is simply what's called a guide RNA to direct this Cas9 complex to the right spot within the genome. There were some initial specificity concerns about using Cas9. Some of the initial papers came out showing how well it worked, and then there was a small group of papers coming out showing the off-target effects of using the CRISPR system. Some of that has now been addressed through improvements in the CRISPR system, and we'll talk a little bit about that. But one of the advantages of CRISPR is really, in order to retarget these things, all you really have to do is supply a different guide RNA. It's the same Cas9 protein derived from bacteria that gets used by different guide RNAs to cause a cut or a nick in the DNA, depending on which version of Cas9 you use. One of the things that we're doing here at Horizon is setting up cell lines that have Cas9 in them prepared for expression. So all you have to do then is to electroporate or transfect in the guide RNA of choice and then you can undertake genome editing right then and there. Just to give you an example of some data and some of the things that we're seeing using CRISPR here, we've done a knockout of the estrogen receptor as an example. We used 5 different guide RNAs in the experiments, and we saw efficiencies that ranged from 1 to 10%. And you will hear reports ranging anywhere up to, I think, 30 or 40%, depending on the guide and the gene and the cell line that's being targeted. There's a great variation in these guide RNA efficiencies. Of the 5 guides that we use, as I say, they range from 1 to 10%, but we also saw differences in their effectiveness in deriving heterozygous versus homozygous events. And all of these things are very difficult to predict. It's more of an empirical check that you need to do to see which guide RNAs are the most effective in your hands. And this is something else that we're driving at to help people with, is launching a program to develop validated guide RNAs and make those available so that customers don't need to actually go sifting through a bunch of guides themselves to find the ones that are most effective. I mentioned there's quite a few recent advances that are coming along in CRISPR. One of the most interesting, I think, as it has to do with genome editing at least, is the idea of using a Nuclease version of Cas9 where only one strand of the DNA then gets cut. This doesn't result in a double-strand break, but if you were to come in with 2 guide RNAs separated slightly in their targets on the DNA, you can in fact introduce a double-strand break, or mimic, I guess you could say, a double-strand break by introducing 2 nicks. And this gets past some of the concerns there were with the off-target effects of the wild-type Cas9. There's quite a few other things, some nuclease-dead Cas9 and other applications that are being developed I won't go into now, but it's a very exciting field and moving very quickly. We're staying right on top of that here at Horizon and trying to evolve with the technology, use it to the best extent that we can, and come up with new products that will help others to use this as well. One of the things I wanted to point out, I talked about how AAV was particularly useful for knock-ins, and it's not to say that a nuclease approach is not particularly useful for knock-ins, but when you're using a nuclease approach for a knock-in, there are quite a few considerations you have to keep in mind. When you introduce, for example, a zinc finger and a double-stranded break, even though you've supplied a donor, you have to take into consideration that there are these multiple outcomes that you see on the slide. For the most part, nothing will happen. Depends on the cutting efficiency, of course, of the nuclease you put in. But at some high ratio, you will start to see effects occurring. And they can be anything from a single allele knockout, a disruption that would occur as if you had not supplied a donor. You can get the double allele knockout. You can get a single allele knockout, a single allele knock-in. You can get a single allele knock-in, which might be what you're looking for, or you could get the double allele knock-in, which is probably the most rare event, but often what people are looking for most. So I'm not trying to knock the technology and say that it doesn't work, but in fact it gets slightly more complex when you introduce a double-strand break. Another thing to keep in mind too, if you're using a nuclease, is that the knock-in efficiency is affected by the distance from the cut site where you want to incorporate the change or changes that you're introducing. And this graph has been used widely to show how that drop-off in efficiency really occurs. You need to be roughly within about 50, 60 base pairs of the cut site in order to get pretty good efficiencies in targeting. One of the things that we've been exploring here at Horizon is the combination of AAV with the nuclease. The data I'm showing here actually involves the use of CRISPR, but it could equally be a zinc finger or a talon. What we've used here is a reporter cell line that harbors a mutant version of GFP. So by supplying a nuclease and a donor that corrects that mutation in GFP, we get glowing cells. So we've supplied the donor here either as a double-stranded plasmid, as a single-stranded oligo, or as the rAAV virus itself. And what we see is the AAV gives a 10 to 50-fold more efficient knock-in percentage than does either the donor plasmid or the single-stranded oligos. So we see quite a bit of utility in the ability to combine these platforms at times. And this is something that you can look for us to be providing kits for in the future as well. So using Genesis, we really can achieve a broad variety of gene editing feats. We can do point mutations, small nucleotide polymorphisms. You see here, for example, a single point mutation. Insertional gene disruption. You can disrupt the coding sequence of a gene using a nuclease, or you can introduce a selectable marker to confirm that all of the cells are carrying that particular knockout. We've done whole gene deletions. You can remove a whole gene, you can remove an exon. You can do long-range deletions. We've been able to take out 100 megabases of a chromosome in one shot. Translocations, we have the EML4 ALK translocation as an example of engineering that was done here in-house. And amplifications is another area where the technology has been applied. So to date, we've done over 70 different parental cell lines, over 500 projects, and that covers 18 different tissue types. The way we're using the technology, just sort of to jump back to the company goals, is to create what we call isogenic human cell lines. So these are cell lines in which we take a particular cell line, introduce a specific mutation, and then provide that pair of cell lines, the parental line and the mutated line, so that people can investigate exactly what are the effects of having achieved just that one change within the genome. And what we've done is to basically model patient genetics in a test tube. We use these then in various screenings. We provide them to customers off the shelf, and we do this as a custom service, generating these isogenic pairs. If any of you are interested in looking at this, you can go online to our website and search these by gene or any of the other parameters that you might want to look through. We also do this, as I mentioned, as a service. It's a comprehensive contract genome editing facility here. We like to think of ourselves as the one-stop genome editing shop. With the application of zinc fingers, with CRISPR, and with rAAV, it gives us all of the tools that we need to achieve really any type of gene editing experiment that somebody is interested in performing. We have extensive experience here. We have hands-on experience with all of these technologies, with over 70 different cell lines. It's something that we can bring our expertise to bear. And as I say, you know, it's always important to us to ask the customer, what is it you're after? What is it you need to accomplish through this genome editing? And then, you know, help them to do that, whether they carry out parts of that themselves and we can assist, or whether we do the whole thing from beginning to end. Those are all options for us. So we've got new products coming out in 2014. There'll be more to see then. I would ask you just to kind of keep your eyes and ears tuned in. We will let you know when those products are available. In the meantime, though, we are here and willing to help anyone basically in any of their genome editing experiments.